Is it good morning or good afternoon? Okay, good morning, ladies and gentlemen. Thank you, Prof. The plant-derived chemotherapeutic that I'll be focusing on this morning is a very well-known drug. It's Taxol. It was isolated from the bark of the Pacific yew tree. Now, ever since its isolation and development, this molecule has played a very important role in the treatment of various types of cancers, especially breast cancer. But what is of interest to us is that recently several groups, including ours, have shown that Taxol possesses anti-angiogenic effects. We showed that it inhibits endothelial cell migration, and it also inhibits angiogenesis in an in vitro assay with much more potency than the established anti-angiogenic 2-methoxyestradiol. And so we embarked on trying to understand the effects of Taxol on angiogenesis. But before I get into that, I just want to touch briefly on the link between angiogenesis and tumor growth. And so several mutations can lead to the development of a neoplasm. But it is often said that that tumor will remain benign unless if it can elaborate its own vascular supply. And indeed, other studies have shown that in response to hypoxia, tumor cells can secrete pro-angiogenic factors. And these pro-angiogenic factors stimulate the development of new blood vessels from a pre-existing microvasculature, a process that is known as angiogenesis. Now, two of the most potent and highly characterized pro-angiogenic factors in the context of angiogenesis are known as basic fibroblast growth factor, or BFGF, which I'll be focusing on today, and also vascular endothelial growth factor, or VEGF. Now, these newly established blood vessels provide nutrients or supply nutrients and thus allow the tumor to grow, and in some instances, to even metastasize. As such, ladies and gentlemen, the targeting of tumor blood vessels represents a very important therapeutic objective. And so, to begin our study, we used endothelioma cells, which are endothelial cells isolated from vascular tumors. We then studied their growth or viability using a crystal violet nuclear staining assay, and the results showed us that there was a dose-dependent inhibition of endothelial cell viability. We then did a study on endothelial cell migration and invasion, and we used BFGF as a chemoattractant. We were using the Excelligent system, and the results showed us that with control cells, they migrated, and on the contrary, the cells that were treated with varying doses of Taxol were inhibited in terms of motility. We then did an in vitro angiogenesis assay. And so what we did was to isolate type 1 collagen from rat tails. We reconstituted it, and we cultured the cells on top of the collagen gel. And so these cells formed a monolayer with a cobblestone appearance. Now, in cultures where we added the pro-angiogenic factor, these cells formed tube-like structures. So to better understand what these tube structures were, we did transmission electron microscopy. And we can see inside the gel, these endothelial cells had coalesced. You can see the nuclei of the endothelial cells. And in the middle, there was a lumen. And so these endothelial cells had formed capillary-like structures. When we added Taxol, at the highest dose, we did not see these capillary-like structures. So we did quantitative analysis, and it showed that there was a dose-dependent inhibition of in vitro angiogenesis. And so we moved on to in vivo studies. So we injected the endothelioma cells to the flanks of mice. We had a control group which did not receive any Taxol. We had the Taxol-treated groups, and we also had a baseline group. Now, with the baseline, there was no tumor induction, just saline. And so in terms of tumor development, there was no tumor growth in the baseline. In the control group, at the end of the study, 100% of the mice developed tumors. And in the 1.2 milligrams per kilogram and 3 milligrams per kilogram dose group, we had a frequency of tumor development of 60% and 40% respectively. And there were no tumors in the group that was treated with 6 milligram per kilogram. And in terms of measuring the size of the tumors, there was, again, a dose-dependent decrease in tumor size based on the dose of Taxol that we administered. We also did protein arrays, and they showed that in those mice that had tumors, we had an increased expression of basic fibroblast growth factor, the pro-angiogenic factor, but we also had another protein, platelet factor 4, which was underexpressed. Now, this factor is an endogenous anti-angiogenic factor. And the expression patterns were reversed in the treated groups. And so to just summarize our observations. So Taxol inhibited endothelial cell growth. It also inhibited endothelial cell migration and invasion, and it inhibited in vitro angiogenesis or capillary morphogenesis. Now, these are very important endothelial cell functions in the process of angiogenesis. So taken together, we can say that in vitro, Taxol inhibited angiogenesis. And so to summarize our observations from in vivo studies, we saw that the decrease in tumor formation was associated with a decreased expression of BFGF, which is a pro-angiogenic factor associated with tumor angiogenesis. We also saw that there was an increase in the expression of PF4, which is an endogenous anti-angiogenic. Therefore, this led to the suppression of angiogenesis. And ladies and gentlemen, it is plausible that the decrease or the tumor growth inhibition that we saw following Taxol treatment could be attributed, at least in part, to its anti-angiogenic effects. And therefore, the role of Taxol in the treatment of these vascular tumors requires further consideration. I thank you for your attention.